Today's lecture is on the cytoskeleton and cell movement. So there's a structure and organization that takes place in cells dealing with actin, myosin, microtubules, etc. And some important things to know, guys, that your cytoskeleton is composed of three main types of protein filaments: actin, microtubules, and intermediate filaments. Actin will polymerize to form actin filaments, or what sometimes are known as microfilaments. And they're flexible fibers about 7 nanometers in length, and they also have, 7 nanometers in diameter and several micrometers in length. So the 3D structure of these guys is pretty cool. Each actin monomer has a tight binding site that will mediate what's called a head-to-toe interaction with two other actin filaments. And if you take a look here, you've got G-actin, and then you've got some dimers that will form, then trimers. Then eventually you're going to have the actin filament with what we call a barbed end and a pointed end. So all the actin monomers are oriented in the same direction, so the actin filaments have the polarity, and it's important in their assembly and in order to establish the direction of myosin movement relative to actin. So nucleation is the first step of actin polymerization, and the dimers and trimers are formed. Then monomers are added to either end. So the actin polymerization is reversible. Filaments can be broken down when necessary. So there's a process called treadmilling, guys, and that's when the barbed end of a filament grows 5 to 10 times faster than the pointed end. Actin is bound to ATP, which associates with the barbed end, and ATP is then hydrolyzed to ADP. So the ADP actin is less tightly bound than ATP actin, and dissociation at the pointed ends. So treadmilling then illustrates the dynamic behavior of actin filaments, and it's critical in regulating actin filaments within the cell. And here's an example of that, guys. If you take a look over here on the pointed end, you've got all of these ADPs. Then eventually, as you get to the barbed end, what you're seeing here is the ATP being bound on this side. So when that hydrolyzes, then the whole process continues like that. So a couple of important proteins, guys. Formins, those will bind ATP actin and nucleate initial polymerization. And profilin, which binds actin monomers and stimulates exchange of bound ADP for ATP. And that's an example then of that. Now, tropomyosins, guys, will actually stabilize actin filaments by binding lengthwise along the groove of the filament. And then capping proteins will stabilize by binding to the barbed or pointed ends. So here's your barbed end. Here's your capping protein. If you look at the pointed end, likewise, you also will have a capping protein. Interesting with the tropomyosin, if you look at it, it's almost like a cord that's wrapped around here, and it's, if you think of a rubber band, that's kind of twisted with that. Cofilin is another important actin binding protein, and basically it severs filaments, so generating new ends that can then be available for polymerization or depolymerization. So again, here's your pointed end, barbed end, cofilin gets added on here, and basically you can break this apart so you can add more or take more away. So actin bundles can be broken into two types: parallel bundles, they're closely spaced, and they have the same polarity. And one important bundling protein, guys, is called fibrin, and that's actually isolated from intestinal villi. Contractile bundles are the second type. They're widely spaced, and they're cross-linked by alpha-actinin dimers. Okay, so increased spacing then between filaments allows the motor protein myosin to interact with the actin filaments. So here's your parallel bundles here, guys. And if you look at contractile, there's more space that's in between here that myosin can then interact with. So in this whole network, guys, the proteins such as filament will form flexible cross-links because you have to have flexibility, guys, for movement. And the filament dimer is a V-shaped molecule with actin-binding domains at the ends. Spectrin is another important protein to know. It's a member of the kelponin family of actin-binding proteins. Ankyrin, again, will link with spectrin, actin, and the plasma membrane will bind these, and it's with a transmembrane protein called band 3. So all of these things will be linked together. So again, you're trying to get the cells to move. Ankyrin's going to link them with spectrin and actin, and then bind up here to the transmembrane protein band 3. A lot of cells have linking proteins. Dystrophin, which is a type of kelponin, is found in muscle cells. And again, it will link actin filaments to transmembrane proteins in the plasma membrane. And beautiful picture here, guys, is some of the different types of stress fibers and what we call focal adhesions that different cells have. These are the stress fibers. They're a pretty cool picture. So a lot of people work with fibroblasts. I work with a lot of these in my time. And fibroblasts will actually attach to a matrix via what are known as integrins. And then the sites of attachment are called focal adhesions. And they're also the sites for stress fiber attachment as well. And those are just large actin bundles. Pseudopodia, guys, are extensions of moderate width and responsible for phagocytosis and the movement of amoeba. And you guys have heard of how amoeba actually move. They form pseudopods, and they eat that way too. Lamellipodia is another type of way cells can move, and they're more broad or sheet-like. And you have extensions at the leading edge of the fibroblasts. And finally, guys, Filopodia, very similar to Lamellipodia, but they are very thin. And they come off the plasma membrane, and they're supported by actin bundles. So, know this. Movement of a cell across the surface proceeds in 3 stages: extension of the leading edge, attachment of the leading edge to the substratum, and retraction of the rear of the cell into the cell body. And here's an example of that. You're extending here, guys, and what you should see is as you move along, these attachments, these little bars here, are attaching, and then once it's moved a certain amount, the back part retracts, and that keeps going on. So again, extension involves branching and polymerization of actin filaments, and then if you inhibit the actin polymerization, that will block the formation of those particular protrusions. So, another important fact to be aware of, guys, is that the retraction at the trailing end involves small GTP binding proteins called ARF and Rho families. And what they do is they regulate breakdown of existing focal adhesions and stimulate endocytosis of the plasma membrane at the trailing edge of the cell. Myosin, I think you guys have all at least heard of that. It's a molecular motor, and it's a protein that converts chemical energy, ATP, into mechanical energy, and that generates force and movement. Myosin 2 is the type in the muscle, and it has 2 heavy chains and 2 pairs of light chains. And here's just an example of that. So muscle contraction then, guys, is triggered by nerve impulses that will stimulate release of calcium from the sarcoplasmic reticulum, and that increased calcium concentration then in the cytosol affects 2 actin filament binding proteins, tropomyosin and troponin. So tropomyosin then will bind lengthwise with the actin filaments, and when the calcium is not there, the tropomyosin-troponin complex blocks the binding of myosin to actin. So binding of calcium to troponin C will shift the whole complex and allow contraction to proceed. Okay, so when you add your calcium here, you're actually exposing all of these little myosin sites on this side over here. And that's just kind of a close-up of the whole complex that we have. Microtubules, rigid hollow rods, they're dynamic and they can undergo continual assembly and disassembly. And again, they function in cell movements and in determining cell shape. Microtubules are made of tubulin, and there's dimers of those, alpha and beta tubulins, which are encoded by genes. Now, another type of tubulin, gamma, is actually found in the centrosome and helps in initiating the microtubule assembly. And again, this greenish one is alpha, the bluish one is the actual beta. So there's a lot of turnover, guys, that will allow the microtubules to be involved in movement and remodeling and cytoskeleton, etc. during mitosis. So different types of drugs have been developed. So I'd like you guys to be aware of those drugs. Two are colchicine and cosamide, and they will affect microtubule assembly and are used to treat cancer. Vincristine and vinblastine are also cancer chemotherapies because they inhibit microtubule polymerization, and that affects rapidly dividing cells. Taxol is the last one, which will actually stabilize the microtubules, and that will block then cell division. So cytoplasmic dynein is another important protein, and what that does is it moves along the microtubules towards the minus end, and it's been previously identified as what's called an axonemal dynein from cilia. Okay, so again, guys, we talked about, or you should be aware of cilia and flagella. They're used for movement. Bacteria have flagella, but not cilia. Eukaryotes do have cilia, as well as flagella, or they can have both, not necessarily do have both. Okay, so the structure of the cilia and flagella are similar. Please know this, that the axoneme consists of microtubules in what's known as a 9+2 pattern. And then there's a central pair surrounded by the 9 outer doublets. Each doublet is complete, a tubule fused to an incomplete B tubule, and this molecule here called nexin links the tubules, and the two arms of the dynein are then attached to each A tubule. So here's an example, guys, of that. So here's your 9 on the outside. So when we say 9 plus 2, that's a beautiful electron micrograph of that, guys, 9 plus 2. Epithelial cells are cells that I think you guys are aware of. Our skin cells are epithelial. And they possess what are known as desmosomes. And these are junctions between adjacent cells. They have keratin filaments that attach to dense protein plaques on the cytoplasmic side. And the attachment is mediated by what's known as desmoplancton. And here's an example of that. So your cells need to be stuck together, and this is basically what the purpose of the desmosome actually is. Hemidesmosomes, guys, are also junctions, and these are found more so in connective tissues. Keratin filaments can also be linked to integrins by different planktons in these particular structures as well. And there's an example of that.